The Human Experiment by Dr. David Carmichael, Chapter 8, Fossil Evidence for Human Origins. You have read about the range of characteristics and complex behaviors that humans share with apes, our closest primate relatives. It is now time to examine some of the differences, especially those anatomical differences that are used to classify the fossil remains that make up the database of human evolution. The general public has often been led to believe that fossil evidence for human evolution is non-existent, or at least not extensive, or equivocal and problematical. Such suggestions are highly misleading, if not outright disingenuous. While there are certainly differences of opinion among scientists about the details of human evolution, the research and debates are based on what is now very substantial collection of fossils, including many that are clearly transitional between apes and humans. Question and answer: are humans evolved from monkeys? No, humans share a common ancestor with the great apes dating back to about 6 or 7 million years ago. Scientists now identify 20 different forms of fossils, hominins. Conroy and Ponser, 2012, 105, spanning the last 6 million years of prehistory. All of these species are represented by at least several specimens, and some are known from more than 500 specimens. There have been 495 specimens of the Australopithecine species recovered from the South African site of Sterkfontein since 1966. The site of Kubi Fora has yielded 129 specimens. Nichols, 1986, estimated that nearly 1,500 Australopithecine fossils representing 200 to 350 individuals were known to science in 1986, and the numbers of sites and fossils discovered has increased dramatically since 1976. Eckert, 2008, suggests that the total number of hominid fossils from Africa and Eurasia may now be around 8,000 specimens. The fossils record of human evolution cannot be dismissed as sparse or questionable. Instead, it provides hard physical evidence of change in the animal forms that preceded our species as well as our nearest primate relatives, the great apes. In fact, instead of saying that humans are related to the great apes, it might be more accurate to say that we are one kind of great ape, the bipedal great apes. We are more similar at the genomic level to chimpanzees and bonobos, and these two species are to gorillas. We are more similar to chimpanzees than mice and rats are to each other. Barkey, 2018. This chapter and the next present in introduction to some of the fossils that demonstrate the key changes in hominin evolution and that define our own ancestry in comparison to apes. Students should be able to identify the key traits that are used to identify and classify animals, fossils or modern, as hominins, and how they differ from the penins, apes. You should also be able to describe why the different genera of early fossil hominins are so classified. That is, in what ways do they differ anatomically, and how do scientists think those distinctions reflect different adaptations? Additional illustrations and detailed descriptions of hominin fossils beyond what those presented in the textbook may be found at the following websites. I will leave this website at the bottom of after the readings because there's 3 of them. Hominins defined bipedal locomotion. A hominin is any member of the taxonomic tribe Hominini which includes modern humans, our ancestors, and a few other closely related prehistoric species. The hominins are often defined in contrast to the taxonomic group known as the Gorillinini and its subfamily Ponginae, orangutans, the subfamily Gorillinae, gorillas, and the tribe Panini, chimpanzees. That is, the modern great apes and their ancestors. Figure 8.1, the hominins may be most simply identified as bipedal primates. Although some of the apes occasionally walk on 2 feet, only the hominins are habitual bipeds. Walking upright is central to the overall adaptation of the hominins, and it influences many details of the skeletal anatomy. In addition to bipedalism, the other defining trait of hominins is non-honing dentation, the lack of overlapping canines. All of the other traits we tend to associate with prehistoric humans, such as tool use and language, developed much later. I'll zone in on that a little closer so that you may see that. Figure 8.1, I'm so sorry that it's jerking like that. General relationship to the hominin lineage to that of the great apes. The anatomical details related to bipedal locomotion and thus to hominin identity can be organized according to 3 major areas of the body: the postcranial skeleton, the skull, and the dentition. The relevant details from each of these areas are presented below in order to best understand them. It will be useful for you to refer to the illustrations of ape and human skeletons. First, let's consider postcranial skeleton, or that part of the anatomy from the neck down. The human skeleton differs in several ways from that of an ape in ways that reflect the overall adaptation to habitual upright bipedal locomotion. Starting with the trunk, note the orientation of gorilla's spine. It is partly horizontal and partly held at a 45-degree angle to the ground, figure 8.2. The pelvis is long and narrow, extending nearly halfway up the back. The center of gravity is below the rib cage and in front of the hips. The weight of the body is both suspended from the pelvis and spine and supported by the forearms in what is referred to as a knuckle walking stance. The rib cage is deep, extending far down from the spine in combination with the abdominal muscles. It forms a sort of hammock within each of which the internal organs are carried. Here's a picture of that, figure 8.2. Figure 8.2, comparison of ape left and human right, skeletal anatomy and posture. Contrast that with the human skeleton, in which the spine is vertical, allowing for upright stance. There are 2 major curves in the hominin spine that reorient the trunk: the lumbar curve at the lower back and a smaller curve in the cervical region or neck. It is no coincidence that these are areas of common pain and injury in humans, for they are compromises in structural arrangements that represent evolution from the ancestral condition to accommodate the upright bipedal stance. The upright stance results in the center of gravity being repositioned over the hips, facilitating weight bearing and balance. Note also that the pelvis is short and wide rather than long and narrow. The human pelvis actually acts as a bucket that supports the internal organs. They are not suspended from our spine and the chest cavity, and rib cage is much shallower, thinner front to back. As a result, in addition, in humans, the limb ratio is different from the apes In humans, the legs are proportionally longer and heavier, and the arms shorter and lighter. The upper legs are angled in from the hip to the knee, in contrast to the bow-legged stance of the apes. This results in the weight of the body being carried through the knee joints rather than between and in front of the knees. The knees themselves are one of the signature traits of hominins, for they are capable of locking at full extension, allowing us to walk efficiently. By straightening the leg with each stride, it was recently discovered that orangutans also have a knee joint capable of locking. Thorpe et al. 2007, Hooper 2007. But that is the only one exception to the rule that locking knees are the characteristics of the hominins. The orangutan seems to have this trait because it also moves with a bipedal motion, but on top of tree limbs rather than on the ground. As a result, the changes in the feet that characterize hominins are not found in orangutans. Those changes involve a reorganization of the foot bones and musculature to form an arched tripod that supports the weight of the body and cushions the foot during walking. Unlike the apes, humans lack a grasping big toe. Instead, the big toe has shifted to align with the other toes, and the ball of the foot is one of the 3 pressure points on the foot. The legs of the tripod that bears our weight. The hominin foot has 2 arches that act as shock absorbers, one that runs front to back under the foot and another that runs from side to side over the instep. None of these details occur in the apes, but all are characteristics of the Australopithecines and later hominins. Figure 8.3, comparison of the size and structure of primate feet, including from left to right, chimpanzee, orangutan, gorilla, and human. Pressure points on the human foot are marked with X symbols. I will focus in on that so that you can see where the X symbols are. Next, compare the way the skull is attached on the knuckle walking ape and the upright walking human. The ape skull actually hangs off the end of the spine, requiring massive neck muscles to hold it up. In contrast, the human skull is balanced atop the spine, so the neck muscles are much smaller. Refer to the figure that compares some of the basic features of the modern human skull with that of a modern gorilla. Figure 8.4. The overall shape of the human skull includes a high rounded cranium portion of the skull containing the brain. With a developed forehead at the front, the face is flat, such that a line drawn from the forehead to the chin is nearly vertical. Supraorbital brow ridges are relatively small or lacking altogether. Comparing the human skull with the gorilla skull, we can see that the overall shape is quite different. The gorilla cranium is relatively smaller and there is no forehead. In fact, there is a constriction of the cranium behind the eye sockets. The gorilla has very large supraorbital brow ridges. The lower portion of the gorilla's face extends quite far forward. This condition is referred to as alveolar prognathism. Prognathism is very prominent in apes, mostly lacking in later hominins, and intermediate in early hominins such as Australopithecines. Using the human skull diagram as a guide, place your fingers above one of your ears about halfway up the side of your head and clench your teeth. You should be able to feel a thin layer of muscles moving under your fingers, and when you do this, if you move your fingers higher while continuing to clench and unclench your teeth, you will reach a point where you no longer feel muscles moving. You are now touching just above the superior temporal line. The temporal line is a small raised area on the skull where your jaw muscles attach. From there, they stretch down behind the zygomate arch, the bone that extends from the cheekbones back to your ears. The muscles then spread out and around the corner and lower edge of mandible or lower jaw. The small size of the temporal line reflects the relatively small jaw muscles it must support. At the back of the skull is a bump just above the hairline called the occipital protuberance. It is usually more pronounced in males than females, again because the size of the muscles that attach there. In this case, the neck muscles. Now compare these features with the analogous areas of the gorilla skull. Notice that there is no temporal line on the side of the ape cranium. Instead, the chewing muscles attach to the sagittal crest a bony ridge extending from front to back along the center line of the top of the skull. Think about that for a minute. While our jaw muscles attach about halfway up the side of our skull, the gorilla has jaw muscles that attach completely above the cranium on a specially adapted bony ridge. These are very large muscles, and their prominent attachment point correspond to the enormous jaw, which makes up nearly a third of the skull. There is also a huge zygomatic bone or arch with large space behind it to accommodate the massive bundle of muscles extending from the sagittal crest to the jaw. Similarly, the rear of the skull lacks an occipital protuberance. Instead, a notable crest extends up the back of the skull from both sides, connecting with the sagittal crest high on the back of the skull. This second set of bony ridges is where the gorilla's enormous neck muscles attach. Finally, let's compare the dentition of apes and hominins. Figure 8.5. The apes illustrated in this case by a chimpanzee retain the older ancestral dental arrangement exhibited by the Miocene apes. The shape of the jaw is rectangular with the side teeth arranged in more or less parallel rows. The largest teeth are those at the front, the incisors and canines, and there are gaps, canine dysautomata, at the front corners of the mouth that accommodate the overlapping canines from the lower jaw, thereby allowing the rest of the teeth to occlude or make contact. The movement of the canines in and out of the gaps actually hones the canines, keeping them sharp and effective for use of agnostic displays. The human dentition is very different in several important ways. The teeth are arranged in a smooth curve and in the shape of a parabolic arch. The side teeth are not in parallel rows but converge toward the front. There are no canine diastemata and no overlapping canines. The teeth are more balanced in size than in the ape condition. That is, the different kinds of teeth are closer in size than in the ape. The largest teeth are those in the rear of the mouth, the molars, and the smallest are the incisors in front. The opposite of the ape condition. Note the jaw of the Australopithecus frensis, aka Lucy, is intermediate or transitional between the apes and humans. There is still a small canine diastema. So the canines of Australopithecus frensis were larger than those of modern humans, but they were non-honing. The tooth rows are not parallel. They have begun shifting towards the parabolic arrangement. Also, unlike the apes, the front teeth are relatively small while the rear teeth are large. It is noteworthy that I'll hone in on these teeth so you can see this, and you can pause the video and read 8.5. Actually, I'll just go ahead and read 8.5 for you. Figure 8.5, comparison of the maxilla upper jaws of chimpanzees, Australopithecus ramidius, and a modern human. The blue arrows points to the canine diastemas, the gap in the tooth row that provides space for the canines in the lower jaw, thus allowing the jaw to close and the teeth to occlude. Note that the canines and the diastemas are large and in the chimpanzees very much reduced in A. ramidius and lacking in modern humans. It is noteworthy that the defining hominin traits, the shift to bipedal locomotion and behavioral dietary changes reflected in the teeth affect many parts of the anatomy. Therefore, they can be inferred from bones representing structures such as hips, knee, feet, spine, and skull. It is not necessary to recover an entire skeleton. It is still possible to identify it as a hominin as long as few key bones are recovered that provide evidence of bipedal locomotion. As important as bipedal locomotion is to the human condition, there is some debate about the timing of and reasons for the beginnings of bipedalism. For many years, the fossils of the Australopithecus afarensis, aka Lucy, provide the earliest evidence of bipedalism among the hominins. The context of those fossils suggested that bipedal locomotion first occurred in a savanna grassland environment, and at least half a dozen hypotheses were developed to explain the beginning of bipedalism. Feeder and Park 2007, 242, Larson 2017, 219. The hunting hypothesis suggested that upright walking freed the hands to use tools to hunt. The vigilance model suggests that hominins would benefit from being able to see over the savanna grassland vegetation to see danger coming from long distances. It has also been suggested that an upright stance would make hominins look taller and more intimidating to predators. Others have suggested potential benefits such as being able to reach higher into trees and bushes to harvest foods, reducing the amount of sunlight hitting the body, allowing for better cooling and the greater efficiency of bipedal walking when compared to knuckle walking. As long as Lucy was considered to be the earliest hominin, most of these ideas could be considered to be plausible explanations of the beginnings of bipedalism. That changed with the discovery of the Ardipithecus ramidius, aka Ardí. See the links provided below for an introduction to the Ardí fossils and their implications for the beginnings of upright walking. Oh my goodness, I can turn the page. Okay, I will leave also these at the bottom of the thing so that you guys may look that up. To summarize, Ardí dates it to about 4.4 million years ago, or a million years earlier than Lucy. Ardí seems to be an evolutionary mosaic containing an unexpected combination of traits. She has a small brain and prehensile feet like the apes, but the shape of her pelvis and the presence of an anterior-inferior iliac spine on the front of the pelvis suggests that she walked upright. Although not as well as Lucy, her arms were long, but her fingers lack the strong metacarpals that would be expected in a knuckle walker. She had reduced non-honing canines. The fossil of plants and other animal species found in the layers in which her bones were recovered clearly indicate she lived in a heavily wooded environment, not a savanna grassland. So, what does all that mean for the potential reasons for the evolution of the bipedal locomotion? If Ardí lived in a dense woodland, as appears to be the case, then her upright stance would probably not result in being able to see danger coming from a long distance. It also would not reduce the amount of sunlight heating the body if Ardí was in shade a lot anyway. Given that she had prehensile feet, Ardí would have been able to easily climb trees to obtain food, so being able to stand up and reach the lower branches would seem to be of little additional use. What does seem likely is that bipedalism would allow Ardí to carry things in the hand, although hunting tools are not known among hominins until a couple million years later. However, it would be useful to carry food, a possibility that the discoverers of Ardí have suggested as an additional potential explanation. Malprovisioning. It has been suggested that because the species has reduced non-honing canines, it could mean that a shift in mating behavior is indicated in this scenario. Males would be able to efficiently travel longer distances, bringing food to their mates, and thereby improve the chances of successfully raising offspring to maturity. Although this idea is by no means fully accepted, it does account for the evidence, and it raises the provocative idea that hominin reproductive success as compared to non-human primates may be at least partly due to the development of pair bonding and sexual division of labor. Hominin taxonomy. The diagram presented in figure 8.6 is simplified presentation of inferred hominin taxonomic relationships. It is simplified because the specific relationships and evolutionary connections among species are still being debated, and because many of the details of that debate are beyond the scope of introductory text. The earliest fossils presently classified as hominins, Sahelanthropus, Chatogenesis, and Ornithogenesis, are not linked with any of the other groups. This is because there are so few fossils of this species that it is difficult to say much about their relationship to other species. Sahelanthropus consists only of cranial elements, and the specimen is interpreted by bipedal and therefore hominin on the basis of the placement of the foramen magnum. The Orrin fossils, if I mispronounce, I'm so sorry, do not include femurs, and that on that basis, it has been determined that this species of bipedal and probably more closely related to Australopithecus than to genus Homo, Richmond and Jungers, 2008. The classification scheme summarized in figure 8.6 has the advantage of focusing on the placement of the better-known species within each of the four main lineages or genera: Ardipithecus, Australopithecus, Paranthropus, and Homo. Although the identity of specific ancestors of humans is still a matter of debate, it is widely accepted that species within the genus Australopithecus gave rise to both the genus Paranthropus and the genus Homo. I'll zone in on this so that you can pause this and take a look at this. I hope it's not blurry. Figure 8.6, the taxonomic guide diagram of hominin species. What does it mean to assign species to a different genre or lineages? The short answer is that researchers have made inferences about the meaning of the differences between the species of genre and have concluded that the genera represent hominins with fundamentally different adaptations. Such analysis depend on the age of the fossils, as well as the identification of ancestral versus derived traits. Ancestral traits are characteristics possessed by organisms due to a shared ancestry. Derived trait are the characteristics that did not exist in ancestral populations, but are instead a result of organisms' more recent adaptation. For example, humans and monkeys both have 5 digits in each hand and foot. However, this particular trait is not indicative of a close relationship because both humans and monkeys have retained the ancestral trait of 5 digits. The general ancient mammalian condition, in contrast, horses and zebras share a derived trait of a single digit, a hoof, relatively rare in mammals and occurring as a specialized adaptation to running on the open plains. Sharing of such derived traits is indicative of close evolutionary relationships. Regarding the hominin traits mentioned above, the chimpanzee and other great apes have retained the ancestral condition of a rectangular dental arch, inherited from ancestral populations of the Miocene apes. Stringer and Andrews, 2005, 120. The ancestral tooth arrangement is also seen in the hominin Australopithecus anemius, but humans show a derived trait of a parabolic dental arch, the loss of honing canines, and the shift to a balanced dentition. One can also think of ancestral traits as representing a more generalized condition and derived traits as those resulting from more specialized adaptations. These terms refer to adaptive potential because evolution is irreversible. A generalized form will have more potential for adaptive flexibility than a specialized one. While the specialized form may be more efficient in the specific context for which it evolved, if conditions change, the specialization may not be useful, and the adaptive potential needed to respond to the changes may have been diminished. Such a scenario seems to have been played out among the 3 best studied hominin genera. Within the hominins, the ancestral condition is represented by the Australopithecines, such as the Australopithecus africanus, figure 8.7. A africanus retains some ape-like traits, such as relatively small brain, pronounced prognathism, large canines, and long forelimbs. Yet the Australopithecines also exhibit certain key human-like traits related to bipedalism, such as a short, wide pelvis, upright spine, locking knee joint, and realigned and arch foot. Such details are best illustrated by the fossils attributed to the Australopithecus africensis, often referred to as Lucy, figure 8.8 and 8.9. In fact, fossilized footprints discovered at the Letola, Tanzania, clearly show that Lucy's species walked upright, 8.10. Here's a close-up. And in comparison of the fossil cast of the Australopithecus africanus with that of the modern chimpanzee. This is the fossil cast of the Australopithecus, aka Lucy. Artist reconstruction of female Australopithecus afarensis. I'm so sorry if I mispronounce these guys. And here is the Laetoli, footprints attributed to the Australopithecus afarensis. So it shows them starting to walk upright. From the combination of traits in the Australopithecines, two different sorts of specializations seem to have developed. The first of these is represented by the genus Paranthropus. The species in this genus all retained the ancestral condition among hominins of bipedal locomotion, but they developed a specialized dietary adaptation, the consumption of small hard food items such as nuts, roots, and seeds. These are evidently processed by simply crushing and grinding them with the teeth, especially the very large molars. As the molars became ever more massive, corresponding changes can be seen in the rest of the skull. Paranthropus forms exhibit prominent sagittal crest, neutral crest, and massive wide zygomat bones to accommodate the large chewing muscles, figure 8.11. These skeletal traits, while resembling those gorillas, were not inherited from a common ancestor with gorillas. Instead, both gorillas were Paranthropus developed massive jaw muscles as part of their feeding adaptations. That the adaptations were very distinct is indicated by the differences in dentation. While gorilla dentation retains the ancestral ape pattern with larger front teeth and honing canines, the Paranthropus teeth are reorganized with reduced canines and much larger molars. The enamel on the molars is thickened, and evidence of heavy pitting and crushing wear has been observed under the microscope. The Paranthropus species were specialized vegetarians and eventually became extinct. Basic characteristics of early hominin fossils are summarized in Table 8.1. The second kind of adaptation involved the continuation of generalized dentation, use of stone tools, and consumption of protein in the form of bone marrow and meat scavenged from carcasses of animals probably killed by carnivores. Fossil casts of the Paranthropus boise skulls showing left broad-faced and the large supraorbital ridge and sagittal crest, large species behind the zygomat arches to accommodate jaw muscles and large molar teeth. So here's a close-up of that. And here is the table. So if you wish to copy this down, this will probably be on the test. FYI. And then here is part of the rest of that, the brain size. Grasslands. Okay. Hominin brain size. The human lineage represented by the genus Homo took an evolutionary track different from that of Paranthropus, evolving from Australopithecus afarensis, A. africanus, or possibly A. gari. Homo habilis, handyman, is the earliest fossil assigned to our genus. Figure 8.12. The earliest indirect evidence of stone tool use is found at the site of Dika in the lower Awash Valley in Ethiopia. Fossil ungulate bones dating to about 3.4 Maya show cut marks and percussion impacts made by stone tools, providing the earliest evidence for meat and marrow consumption by hominins. McFerrin et al. 2010. Given the age of the site, this meat scavenging behavior can be attributed to the Australopithecus africanus, the only hominin species known from that region at the time. This pattern of behavior becomes more common at later sites, which attributed to the Homo habilis, a species that exhibits a significant increase in brain size over earlier hominins and is believed to have been a consistent maker and user of simple stone tools. Whichever foods H. habilis was eating, they were processed partly with stone tools, eliminating the need for the development of large jaws and massive teeth. It seems the stone tools were used to assist early members of the genus Homo in their scavenging of meat and bone marrow. Animal bones at H. habilis sites are often crushed and split, apparently for the extraction of bone marrow. Some bones of game animals, such as antelope, show microscopic cuts that indicate the animal was first gnawed by a large carnivore, such as a hyena, and then processed by a hominin with a stone tool. Thus, the evidence suggests that the human's lineage reflects a specialization involving increasingly large brains and increasing use of stone tools. Recent research indicates that these two developments are closely interconnected. Fossil cats of Homo habilis skulls. Oh, fossil casts. I'm so sorry. Our ancestors have been consuming increasing quantities of meat for at least 2 million years. Man, 2000. The shift to the consumption of meat, densely concentrated sources of calories and energy, is understood to have been a key element in the evolution of genus Homo. Kaplan et al. 2161, especially changes in the brain that led to what we identify as intelligence. The result of this shift to meat consumption on brain size are illustrated in the graph reproduced below. All but one of the members of the genus Homo have larger brain volumes than the Australopithecines, and the distinction increases over time. It seems that the increasing consumption of lean meat by our hominin ancestors was both necessitated by and allowed for the increase in brain size that has been hallmark of our lineage. There's that. That is the end of chapter 8 for Dr. Carmichael's book. Thank you.